All right. It's time for our next presentation. Please allow me to welcome Tianhong Zhao, Mo Chen, and Yubo Zhao from Tsinghua University in China from Zhaigong Zhang's group. Tianhong Zhao is a first-year master's student focusing on single-cell transcriptomics and foundation models for life sciences. Mo Chen is a first-year PhD student working on spatial transcriptomics and foundation models for life sciences. And Yubo Zhao is an incoming PhD student focusing on foundation models for single-cell research. Thank you very much that you could join us. And for the audience, they presented us with a pre-recorded talk just to avoid any technical glitches, but do note that they're present as part of the panel. So if you have any questions, feel free to put them in the chat, and there's going to be a Q&A toward the end where they'd be happy to answer any of your questions. So in just a second, I'm going to play the pre-recorded talk.
Hello everyone, I'm Zhao Yubo from Xuegong Lab. We are glad to introduce our works, IC Foundation, IC Mulan, Rome, and GEST, Powerful Biology Foundation Models. The title of our today's talk is Multiscale Foundation Models. First, please let me introduce who we are. Our tutor is Xuegong Zhang professor from Tsinghua University, and there are 25 members in our group who are interested in the area of life science and informatics. Then let's begin our talk. In biological research, one of the greatest challenges is understanding how different biological scales work together as an integrated system, from molecule to entire organ systems. These scales do not operate in isolation, but rather interact in ways that are both complex and dynamic. At the molecular level, processes like protein-protein interactions and gene mutations form the building blocks of biological functions. However, their impact often depends on how they are interpreted and executed within the cellular environment. On the cellular scale, cytotransduction pathways and cellular dynamics translate molecular information into actionable responses. These processes are interconnected and must be understood in the context of their downstream effects. Finally, at the organ and system scales, the challenge becomes even more apparent. Systems like the nervous system rely on tightly coordinated communications between cells and molecules to maintain homeostasis. Here, the lack of models that connect cellular functions to organ-level outcomes limits our ability to make accurate predictions. Biological research in models that bridge these scales, providing a framework for exploring and predicting the intricate relationships within living systems. Another major challenge in biological research is from the explosion of multimodal data generated by modern bioinformatics technologies. Advanced tools like sequencing, mass spectrometry, and medical imaging provide vast and detailed datasets across molecular, cellular, and systemic levels. However, integrating these diverse modalities into a cohesive framework for analysis and modeling remains a significant hurdle. Data from different scales and modalities often lack standardization, making cross-platform comparisons and unification complex. Furthermore, the biological patterns researchers seek are frequently buried within layers of noise, requiring advanced algorithms to extract meaningful insights. These challenges require innovative approaches to data preprocessing, integration, and interpretation. To fully harness the potential of multimodal data, it is important to address the limitation of traditional methods. These approaches often rely heavily on expert knowledge and manual correction, making it difficult to manage the complexity and volume of large-scale datasets generated by modern bioinformatic technologies. Additionally, traditional statistical models are primarily designed to capture local relationships within the data, leaving global and cross-scale interactions underexplored. To solve this problem, we need tools that can handle multi-scale, multimodal data comprehensively and efficiently. Foundation models represent a transformative approach to artificial intelligence, characterized by their ability to generalize across a wide range of tasks using a single, large-scale model. The term foundation model was introduced in the seminal paper on the opportunities and risks of foundation models in 2021, which outlined their defining features and potential applications. Foundation models exhibit the property of homogeneization. This means that a single, versatile model can serve as the backbone for diverse tasks across domains, leading to a unified approach in artificial intelligence. By consolidating capabilities into one model architecture, foundation models simplify the development pipeline, reducing the need to create task-specific models from scratch. At their core, foundation models are built on self-supervised learning, enabling them to learn from vast amounts of unlabeled data. This training paradigm allows the models to acquire a deep understanding of patterns and representations, which can then be fine-tuned for specific downstream tasks. Their scale, often involving billions of parameters, contributes to their remarkable performance, as they can capture intricate relationships in data that smaller models may overlook. A key property of foundation models is their versatility. Unlike traditional models designed for singular tasks, foundation models like GPT are pre-trained once and can adapt to various applications, including text generation, text classification. An example of biological foundation model is SCI Foundation. It's pre-trained on single-cell data using a mask token prediction strategy. It enables multi-task applications such as cell type annotation, perturbation effect prediction, and regulatory relationship discovery, showcasing the versatility of foundation models in advancing single-cell biology research. Another question we care about is why foundation models can work. First, foundation models are fundamentally enabled by self-supervising learning. It's a paradigm where the pre-training task is automatically derived from unannotated data, driving the model to learn representations that are richer and more versatile compared to traditional supervised methods. This makes foundation model can use a wide range of data. Modern internet-scale datasets encompass diverse modalities often without explicit annotations. Self-supervised algorithms are designed to exploit the unique properties of each data type, crafting tasks like predicting mask words in text, reconstructing parts of images, or completing sequences in an audio recordings. These tasks allow model to learn universal features that generalize across domains, unlocking insights from vast, unstructured datasets. A second critical aspect is domain completeness. Effective self-supervised tasks are crafted to require the model to develop broad capabilities that are transferable to various downstream applications. For instance, training a language model to predict the next word in a sentence inherently teaches it to understand context, sentiment, grammar, and even word knowledge. Similarly, foundation models in single-cell data are trained to predict masking expression values in a cell. This can teach the model the knowledge about biological process and the meaning of genes. Comparing with traditional supervised tasks, which are often trained on task-specific skills, limits the model's adaptability. Like the single-cell model for annotate, the cell type cannot learn the biological process knowledge. While self-supervised learning enables foundation models to acquire broad and general capabilities, fine-tuning tailors these capabilities to specific tasks or domains. During fine-tuning, the pre-trained model is exposed to a smaller, task-specific dataset with labeled examples, allowing it to adapt its general knowledge to particular requirements. Fine-tuning teaches the model domain-specific knowledge that may not be fully learned during pre-training. This is computationally efficient, as it leverages the foundational knowledge already embedded in the pre-trained model, requiring significantly fewer resources compared to training a model from scratch. In addition, foundation models can integrate multi-model data by learning unified representations across diverse data types, such as text, images, audio, and more. So, pre-training on vast datasets containing paired or aligned modalities, like image-text pairs. They capture relationships between these forms of information. Techniques like cross-attention layers or joint embedding space enable the model to align features from different modalities, allowing a seamless integration. These unified representations accelerate downstream tasks like disease diagnosing.
Hello, I'm Tianhong Zhou. As we have seen, foundation models have achieved remarkable success, especially in the field of natural language processing. Their ability to understand and generate complex patterns have revolutionized many domains. Building on this success, we are now exploring how foundation models can be adapted and applied to multi-scale biological scenarios, where they can provide insights into complex biological data at different levels of analysis. From cellular to organ-wide scales, our team has currently developed 4 distinct foundation models: Jest, Room, Single-Cell Mulan, and Single-Cell Foundation. Each of these models focuses on tasks at different scales of biological analysis and has demonstrated exceptional performance in their respective areas. Here, I will introduce their method and applications. Let me introduce Single-Cell Foundation first. This is a foundational model for single-cell transcriptomics. We trained it using 50 million single-cell datasets containing 20,000 genes. These datasets scan over 100 tissues and various organs, with tumor data specifically distinguished. This allows the model to learn gene-to-gene relationships across different scales. Single-cell foundation is based on the extremal gene framework. This is a model designed for single-cell RNA sequencing data. It uses an embedding module to turn gene expression data into high-dimensional vectors that the model can understand. Its asymmetric architecture is optimized for the sparse nature of single-cell RNA data. This design overcomes the limitation of traditional transformers, which struggle with long gene expression. To train the model, we designed a sequencing depth recovery task. In this task, the model receives a gene expression sequence that has been downsampled using Bayesian sampling with part of the sequence masked. The goal of the task is for the model to predict the corresponding longer original gene expression sequence. During the training process, we update the model's parameters by calculating the loss between the predicted sequence and the original sequence. This method helps the model learn how to recover missing gene expression information, thereby better understanding the patterns of gene expression and enhancing its ability to predict gene sequences. Through this training task, the model is able to effectively infer and recover missing parts of the gene expression data, even when the data is incomplete. This allows the model to both understand gene expression and learn how to enhance gene sequences. After training, single-cell foundation can understand both gene-to-gene and gene-to-cell relationships. The model treats genes as words and entire cells as sentences, allowing it to capture the complex interaction between genes within a cell and between cells. This approach enables single-cell foundation to generate enhanced gene expression outputs for each cell. These improved predictions help address missing or noisy data and provide more accurate gene expression profiles. The enhanced output can then be applied to various downstream tasks, such as clustering, cell annotation, and perturbation prediction. First, let's look at how single-cell foundation performs in clustering. The metrics show that as we increase the gene sequence length generated by the model, the clustering performance improves. This indicates that single-cell foundation's deeper gene sequences are better at capturing cellular heterogeneity. Longer sequences provide more detailed gene expression information, allowing the model to better distinguish between different cell types and states. When we map the clustering results to the reference clustering plot, the UMAP visualization clearly shows that single-cell foundation performs better than the SOTA model, with more distinct and well-defined clustering. This demonstrates the model's ability to capture subtle differences between cells, making it more effectively in clustering tasks. Then, we selected data with the sequencing dips of around 500 and applied dips enhancement. As shown in the violin chart on the right, the enhanced dips is mainly concentrated around 2000, indicating that single-cell foundation is effective in recovering deeper sequencing data. This dips enhancement helps improving the overall gene expression data, enabling the model to capture more detailed and accurate information. Further, the UMAP visualization reveals that data generated by single-cell foundation significantly improves the separation of cell types, such as CD14 and CD34. These two cell types will have similar gene expression profiles in the original data, are better distinguished after the sequencing dips enhancement. This demonstrates that single-cell foundation not only recovers missing data, but also enhances the model's ability to identify subtle differences between cells. Single-cell foundation can also be applied to various downstream tasks, such as drug cancer effective concentration prediction and drug sensitivity single-cell classification. For example, in the first task, drug embeddings are generated using a graph neural network, while bulk embeddings are generated using single-cell foundation. These embeddings are then integrated into a predicted model to estimate the IC50 value, which represents the effective concentration of the drug against cancer cells. So, as long as the embedding generation capability of single-cell foundation can be properly utilized, downstream tasks can be easily carried out. Next, I will introduce another foundation model for single-cell analysis, single-cell Mulan. The training data for single-cell Mulan comes from the Human Ensemble Cell Atlas, with HEC for short. This is a comprehensive database developed by our team. This database consists of 10 million single-cell data points, making it a valuable resource for large-scale single-cell research. HEC serves as a platform for integrating vast amounts of scattered in single-cell data into a unified, structured giant table, which helps streamline the analysis of complex biological data. The training data of single-cell Mulan includes single-cell data from a wide range of tissue and organ scales, ensuring that the model is exposed to diverse biological contexts. The dataset incorporates 20,000 genes and 160 gigabytes of natural language corpora, allowing single-cell Mulan to leverage both genomic data and textual information for a more holistic understanding of cellular processes. This rich and varied data enables single-cell Mulan to perform well across various tasks. The single-cell Mulan model framework is built on a powerful architecture with 3,068 million parameters. This large-scale model allows single-cell Mulan to handle the complexity of single-cell data with high capacity and precision. One of the key innovations of the framework is its ability to unify both gene expression and meta-attributes of cells by modeling them as C-sentences. These C-sentences encapsulate the genetic information and cell-specific metadata into a cohesive representation, providing the model with a more integrated view of the data. Once modeled as C-sentences, the data is converted into embeddings, which are high-dimensional vectors that capture both fine-grained details and broader contextual information. This enables the model to understand the intricate relationships between genes, cells, and the biological processes it represents. Through these embeddings, single-cell Mulan is capable of not only identifying cell types and gene expression patterns, but also capturing the underlying biological context, such as tissue or organ-specific characteristics, allowing for more accurate and insightful analysis of single-cell data. During training, we designed 3 key tasks for single-cell Mulan: conditional cell generation, cell type annotation, and meta-attribute prediction. As shown on the left, the input consists of a C-sentence, which includes all information except the output we aim to generate or predict. The output varies depending on the task. In conditional cell generation, the model generates cell profiles based on given conditions, such as gene expression. For cell type annotation, it aligns specific cell types to cells based on their characteristics. Meta-attribute prediction involves predicting additional attributes, like tissue type or developmental stage. By training with these tasks, single-cell Mulan becomes capable of handling a wide range of tasks involving both cell-related information and meta-information, making it suitable for single-cell analysis in various biological contexts. Next, let's take a look at single-cell Mulan's performance across various tasks. First, in the task of integrating single-cell RNA data from multiple batches, single-cell Mulan demonstrates both fast and accurate integration. According to the metrics, single-cell Mulan achieves a high performance level on large datasets and achieves a leading level on COVID-19 datasets. Notably, these datasets consist entirely of unseen samples that single-cell Mulan has never encountered during training. Moreover, in terms of generation time, single-cell Mulan significantly outperforms single-cell GPT and Silrat, making it much faster. Single-cell Mulan also performs well in conditional generation tasks. In this task, we input the metadata of the original data type into single-cell Mulan. By comparing the UMAP visualizations of the original and generated data, we can observe that single-cell RNA data generated by single-cell Mulan closely matches the real data in terms of the UMAP distribution of cells from different organs and cell types. In annotation testing, when the single-cell data comes from organs that the model has encountered before, single-cell Mulan outperforms current SOTA models across 3 classification metrics in 3 dataset application scenarios, demonstrating its powerful annotation capability. When annotating data from unseen organs, Single-cell Mulan performs slightly less effectively with limited fine-tuning data, but it is highly sensitive to the amount of fine-tuning data. As the sample size increases, its performance improves significantly. This highlights single-cell Mulan's strong scalability and flexibility in handling different tasks, especially when sufficient fine-tuning data is available. Finally, let's take a look at single-cell Mulan's performance in perturbation prediction. For the gene perturbation prediction task, the model's input includes the cell's initial state and the target genes to be perturbed. The model's output generates a new gene expression profile after perturbation, reflecting the cell's altered state. In the figure below, we observe the trends of cells shifting in two directions as GITA1 and SB1 genes are progressively overexpressed. The perturbation trajectories show that overexpression of GITA1 makes cells shift closer to the MEP type, while overexpression of SB1 makes cells shift closer to the LMPP type. This demonstrates the model's deep understanding of cellular and background information and gene regulatory networks, as well as its strong generative ability in predicting gene expression changes.
Now, let's move on to the tissue scale. To capture the intricacies of tissues in their original shape and structure, we mainly rely on two types of data: pathology images and spatial transcriptomics. Images provide detailed morphological information and are widely used in tasks such as tumor diagnosis, while spatial transcriptomics data enables us to understand the spatial distribution of gene expression across the tissue, providing insights that go beyond morphology. Let's dive deeper into the models that work at this scale, starting with ROM, which is a transformer-based image foundation model designed for glioma diagnosis. ROM leverages whole slide images, which contain very high-resolution details about tissue morphology. The model breaks down images into smaller segments called patches and treats images as sequences of patches, similar to how language models process sentences, and use special design transformer architecture to extract image features from whole slide images at different resolution scales. By pre-training on large-scale whole slide image datasets, ROM is able to perform gliomas diagnosis, molecular status prediction, and many more tasks, which we will introduce in detail in the coming slides. Here we demonstrate ROM's ability of glioma diagnosis on two critical subtasks: glioma detection and glioma subtyping. The glioma detection task is to distinguish between normal tissue, gliosis, and tumor, while the glioma subtyping is to classify glioma into three known subtypes. Here we use classification metrics to evaluate ROM's performance, including AUC, accuracy, precision, recall, and F1 score from left to the middle. It is apparent that ROM achieves superior performance on all of these metrics, leading a lot from the baseline methods. We also show the confusion matrices on the right to give an intuitive visualization of the prediction results of ROM. We also evaluate ROM in predicting molecular features closely associated with glioma diagnosis, which includes IDH mutation and MGMT promoter methylation. Again, these are supervised classification tasks, so we use the same metrics to evaluate and visualize the performance of the model. As is shown, the AUC in both molecular feature prediction is around 0.9 and clearly outperforms all baseline methods. The superiority performance is consistent with other metrics, as shown in the middle and the right of the slide. These results indicate that ROM has successfully captured effective morphological features from pre-training on large-scale pathology images and can effectively predict molecular characteristics. Here, we present ROM's performance on an independent external test dataset. The dataset used here comes from TCGA, short for the Cancer Genome Atlas, comprising 618 whole slide images collected under different imaging conditions and from diverse institutions. This external dataset introduces additional challenges due to variations in slide processing and imaging equipment, which test the model's ability to generalize beyond the data it was trained on. However, the performance is still quite promising, while there is a slight decrease in performance compared to the in-house test results mentioned before. This is intuitive as this is an external test dataset. ROM can still reach an AUC under 0.9 and consistently outperforms all the baselines on this external test dataset. These results validate ROM's robustness and adaptability, showing that it can generalize to data from completely different sources. This is a critical step in ensuring that ROM is not only effective on controlled datasets, but also applicable in real-world clinical scenarios across diverse institutions. This slide highlights how ROM serves as an auxiliary diagnostic tool, showcasing its potential to support pathologists in clinical practice. First, we directly compared ROM's diagnostic performance to that of 5 practicing pathologists with varying levels of experience, 1 junior, 2 mid-career, and 2 senior, and show the results on the left. As you can see, ROM consistently outperforms 4 out of 5 pathologists in glioma detection and subtyping tasks, and is comparable to the best senior pathologist. We also explored how ROM could enhance the performance of less experienced pathologists when used as an auxiliary diagnostic tool. This is another highlight of this model, as it can not only provide prediction results, but also visualize its attention scores on the image for better interpretability of the results. We use this visualization to assist junior and mid-level pathologists, and witness a significant improvement in their prediction performance. This highlights ROM's potential to elevate diagnostic standards in regions with limited access to skilled professionals. Taking a step further, ROM isn't just assisting diagnosis, it is actually advancing our understanding of gliomas. The strong performance of ROM in predicting molecular features suggests that it captures critical morphological patterns related to that. To explore this, we collaborated with 2 senior pathologists with over 15 years of clinical experience. Together, we analyzed the high attention regions identified by ROM in pathological slides with IDH mutations. We first provided heat maps highlighting high attention regions in each slide. These visualizations were analyzed by the pathologists through the same online platform used for auxiliary diagnosis, as demonstrated on the left. From high attention regions, the pathologists identified 2 distinct morphological features associated with gliomas harboring IDH mutations. These new biomarkers allows for the determination of IDH mutation status without the need for DNA sequencing, which is especially valuable in settings where molecular testing is unavailable. Incorporating these features into diagnostic standards could enhance accuracy and may improve patient prognosis. Now, let's shift focus to spatial transcriptomics and introduce GUEST, which is an ongoing project of our lab. This is a specially designed generative pre-trained transformer for generating spatial transcriptomics data. This model leverages a specially designed attention mechanism to model cellular spatial correlation and iteratively fills unseen region with predicted gene expression. The newly predicted gene expression is then treated as ground truth and is used to further predict the unseen region nearby. This fashion is known as the autoregressive generative fashion, which is exactly what ChatGPT do with natural language. In this process, the model is forced to understand the spatial context of gene expression in order to get accurate generation results. And with a pre-trained model, we can then perform some interesting tasks on spatial transcriptomics data. One possible scenario is to predict spatial perturbation. As is mentioned before, genes are somehow correlated, and if you perturb one, the other may change accordingly, which is an important biological process in our body. Prediction of spatial perturbations allows us to study how cells respond to changes in their spatial context, enabling insights that are otherwise difficult to obtain experimentally. The in silico spatial perturbation includes manipulation of the gene expression in some region of interest, and predict the gene expression of the region around it while the generation process. In the experiments, the guessed model is able to accurately predict gene expression and classify differentially expressed genes. This is still ongoing, and we are also exploring other application scenarios of this autoregressive spatial generation paradigm. Throughout this presentation, we've explored a suite of foundation models that operate at different biological scales, each contributing to our understanding of life in unique ways. These models demonstrate the power of foundation models tailored to specific scales, providing tools to understand the complexities of life at multiple levels. Together, they form a multi-scale framework for studying life sciences. But what if we could take this a step further? The ultimate vision is to integrate these scale-specific models into a cross-scale foundation model, uniting insights from genes, cells, tissues, organs, and even the entire body. Such a model could serve as the next paradigm in life sciences, a digital framework to simulate, predict, and understand life as a cohesive system. This cross-scale foundation model could revolutionize research, bringing us closer to a digital representation of life itself. This is not just a distant dream. Our lab and many other fellow researchers together have already begun this journey. By combining multi-scale data and leveraging advances in generative modeling, we aim to create a unified model that could transform life sciences and medicine, enabling breakthroughs that were once unimaginable. And that is all. Thank you very much. Any questions?
Great talk. Thank you, Tianhong, Yubo, and Mo, that you're able to present. I do have a couple of questions, but I was wondering if anybody else on the panel or in the audience would like to ask any questions. All right. So I was wondering what, maybe I missed this, but what are some of the key differences between the SC Foundation and the SC Milan models?
Hello, Yash, can you hear me?
Yeah.
Okay. The SC Foundation focuses on the gene embedding enhancement, but the single cell Mulan just he focus more on the downstream tasks like the conditional generation and the cell type annotation. Yes, in the single cell foundation generate the embedding you can use to do many things. For example, you can recover the sequence dips. Yes, it's just generation the enhancement embedding that you can utilize to do everything.
Right. So would you say that SC Foundation then becomes like step one before using like SC Mulan, or is that like a completely different thing?
It's a completely different thing
Okay.
because yes, the SC Foundation it is the first our trial to do the gene information, and the SC Mulan is the second trial. Yes.
Right. Okay. I was also wondering, what do you see as some of like the big challenges and limitations in the usage of these foundation models for this kind of single cell analysis?
I think in the current information we might face is the metadata. For example, the information of tissues and organs. Of the current path we use now is the we view the metadata as a token to for model train, but it can't utilize the whole information of the metadata because we are now view the metadata and the gene information as the sentence. It is also the traditional process of natural language. So we might discover how we can do to the gene information, not just you utilize the process of natural language processing.
Right. And for the models where you use like a lot of imaging data, how do you face challenges with like, not finding sufficient amount of like annotated data sets with the imaging? And also like, how do you deal with sort of just lack of like labeled imaging data sets?
Okay, can you hear me?
Yeah.
Okay, that is a good question. For the annotated data set, it is indeed very sparse. So as you can see in the training process of the foundation model, actually it uses a self-supervised training paradigm, which actually do not requires many annotated data sets. It just like supervise itself and use the image itself as the label. If you're really interested in this, maybe you can refer to uh, how the transformer-based models are trained on the maybe first you in the natural language processing, and then you can understand how the image-based foundation model are trained like a self-supervised paradigm. And actually, the self-supervised paradigm is to get a good representation of the image. And with that representation, you can do many things like downstream analysis, like whatsoever. Yeah, I think that will answer the question.
Right. And I was also wondering, so with large language models, like hallucination is like a big issue. So in your work with the foundation models that you're building, how do you sort of see that? How do you deal with sort of this hallucination problem with these large language models?
Yeah, it is another good question. Actually, it is a general problem, like the hallucination with the large scale models. I think.
Right. Yeah. Do you all, I was also wondering, like, with all of these different sort of, foundation models and sort of this cross-organ foundation model, do you see them also being translated, sort of beyond kind of research problems and going into like clinical practices? And if so, like, what are the, like, some of the challenges that you see over there?
I think one big challenge is what you just said is the hallucination problem. Is that is about that is a question about trust. It's like how to what extent do you trust the results of the given by the foundation model? Uh, Although we can evaluate it on some benchmark data sets or something else, but in real application scenarios, I don't think the foundation model will serve as a single decision-making tools. It will just serve as some auxiliary tools. I think, like what I said in the when we're introducing room. I think, yes, especially in the real clinical case, where when you make a decision, when you make some critical decisions, I think.
Mhm. All right. Does anybody else in the panel or the audience have any questions? If so, feel free to put them in chat.
Yash, can you hear me?
Yeah.
Okay, I have a question. Great talk, guys. I was just wondering if you foresee the prospects of a kind of unified foundation model, or if you've heard, I'm curious. I don't have a lot of expertise in this area, so as a layperson, are there standards being developed for what would constitute a good foundational model that would allow you to maybe unify these models? Because some of them, different models have different strengths, right? So I'm just wondering if there is interest or movement towards unifying them, because we see foundational models in different places.
Hello.
Yes.
Can you hear me? Yeah.
Mhm.
I'm not sure if I quite understand what is said by unifying the foundation models. Do you mean that maybe we can utilize the power of all foundation models and make them into a better foundation model, better than any single one? I'm not quite sure if you mean that. Can you clarify that?
So let's say we're working on the Human Reference Atlas, right? And so we're working across the different scales, so single cell level, but also anatomical structures, organs, and the entire body, right? So imagine, hypothetically, I know this is kind of far out, but imagine a foundational model for the human body, let's say, right? Or a cross-scale foundational model, that sort of thing. Is that doable now? Is that because I've seen different, and again, I apologize if it doesn't make too much sense. I'm not an expert in this area, but I'm just
Okay.
curious. Yeah.
I get it. I get it. Actually, uh, it may not. I think it may not be an idea that will be implemented so soon, but we are trying. Many others are trying. The biggest problem in this cross-scale foundation in building this cross-scale foundation model is that we do not have that much paired data set. Let's say that if you have some cross-scale data that's sampled from one human body and then they just from the one instance, and maybe they contain some common information and which can be used for training the model. But in the real cases, we do not have that much this kind of data, and that will pose a problem in training the model. And we have proposed some technical solutions to that. For example, maybe we can introduce some knowledge to the model architectures, and that's just one solution. And yes, I think it is not that doable now currently, but it will be implemented sooner or later. I think it is a trend in the foundation in researching foundation models.
Because our team, and I mean, Yash can speak to this better than I can, but our team has been working on building those knowledge graphs and ontology models for the human body. So it seems like that would help bring some structure into the training, right?
Yeah,
these
yeah.
models.
Actually, our team has also think about the knowledge graph solution, and we have a small team trying on it. Yeah, maybe we can contact later.
Thank you very much.
Yeah.
All right. I did have one last question, and I guess it kind of connects back to the hallucination problem, which is sort of in practice, I think a lot of sort of biologists, especially when they, see these tools that we, in the machine learning world we develop, one of the issues that they have is sort of the confidence problem, which is, if the model is predicting something, again, like how do you tell that that's actually like a true prediction in like a real world scenario? And so I was wondering, like, if your lab is also working, or if you've thought about this in terms of providing some sort of like a confidence, to the predictions themselves. Now, again, like providing a confidence value also is not like a perfect thing, and there are like limitations there as well. But I was wondering, like, if that's something also that you're thinking in terms of like building these models.
Do you mean that the model should give a confidence score when giving the output?
Yeah, in terms of like, when you're like making some prediction, also giving some sort of like confidence value of like how confident the model is that it's actually like a true prediction.
Okay, that I think the current models may not have such capacity, and I actually it is a very big challenge. I think, even for the large language models, which we maybe can explain better with the output, like he actually can talk, but maybe he will give you some confidence scores. But there's another problem. What is confidence score of the confidence score he gets?
Yeah.
I think this is another open problem, I think.
Mhm. Yep. All right. Okay. Kari has a question. Let's see.
Hi, good morning, good afternoon. Good to see you. It's really nice.
So I just promoted her to panelist. Maybe that's why she went away.
Yeah.
Okay.
It like restarts. Kari, you're muted.
Good morning, good afternoon. It's really nice to have this work presented. We had Jur Levkovich presenting earlier in the 24-hour event, and he also has a multiscale models that you can ultimately use to interlink data from the biomolecular level to the cell level to the multicell level. And I would love to see a comparison of your work with the work that is performed by his team. And one way to do it could be to use the rich data that now becomes available via HubMap and many other data portals to see if we can do a head-to-head comparison of these approaches. And I wonder if you are interested, available to do that.
It's a very good proposal, and I think Maybe we can contact later about the details of this such comparison. Actually, I'm very interested about the data set that you just mentioned. If I understood, maybe we'll serve as a benchmark data set later. Yes, maybe we can try. I'm not sure. Yeah.
So maybe you have already looked at this, but we have a recent paper that just got accepted for Nature Methods as a resource. And in that paper, we have downloaded data from 4 different data portals, about 10,000 data sets. And these are single-cell RNA-seq, but also spatially explicit data sets. And I put the link in the chat. And then, as you might have seen, the Human Reference Atlas effort also has many APIs that you can use to get to that data. So you don't have to download all that data again. You can access it in a harmonized fashion. And that might make it much easier to do these algorithm comparisons.
Okay, I understand. Thank you very much for the link.
Maybe I put the link to the APIs in here as well. And we just had a new release yesterday on Friday. So that's 2 days ago now. 24 hours is long. So I put that link into the chat also. So you could use the HRA API. You could also use the client APIs to get to some of that data. And if you have questions, please let us know. I think as there are many teams around the globe starting to analyze single cell data across scales, it will be really, really interesting to have a central repository for some of the data, to have a platform where we can compare these different algorithms, and then to also have a forum to present back what just happened. And so we will have another 24-hour event next year, and there will be many AI algorithms presented at that event. And so it would be wonderful to have your team come back yet again and present. And I think we will absolutely try to get some of the other teams back, which are using massively large language models and other forms of AI, to see what we can do together, human intelligence and machine intelligence. So thank you again for presenting here. I think your work is highly relevant. And we want not just US and European researchers to work on this, but we need an atlas that's relevant for all human beings on this planet. And it requires that many people around this planet get to have a say and get to help prioritize data sets and algorithms. And so it's really important that this event goes around the globe so that everybody can present in their respective time zone.